Hello, everyone. Thank you for joining us for our first ISHR Mid-Career Investigator webinar of the year. We are very excited to have two wonderful presenters for you today. We have Dr. Onur Kanisicak from The Ohio State University and Dr. Emily Tsai from Columbia University. And we also have two early career moderators, Eric Blackwood from the University of Utah, as well as Glynis Gary from UT Southwestern. But before we get started with the webinars themselves, um, just have a few bits of information I wanted to convey to everyone. So the first is that, sorry, my slides are, there we go. The first is just about our Mid-Career Investigator Committee. So this is a committee that was formed to support mid-career investigators at what we feel is a very crucial stage of their careers. The members of our committee range from senior assistant professors to even those who are newly promoted full professors, but typically people who are 8 or more years beyond their first faculty appointment. So as a committee, we're committed to supporting all of our mid-career investigators within the ISHR, and we plan a lot of mid-career investigator symposia at section meetings. We also take on initiatives like this webinar series, and we plan to hold this on a quarterly basis throughout the year. So please be on the lookout for our next webinar in June. And certainly if you're interested in becoming a part of this committee, or if you would like to present in this webinar series, please feel free to reach out to any of us. Additionally, I wanted to let everybody know that the North American section has a wonderful website. It's still relatively new. It went live a couple of years ago. And from the website, you can do all kinds of great things like renew your membership. You can find more information about our Mid-Career Investigator Committee. And there's also information on there about upcoming section meetings. So on that note, I wanted to remind everyone that there is an upcoming 2025 ISHR World Congress meeting in Nara, Japan. That will be taking place in May. And meeting registration and abstract submission is still open. But the deadline for abstracts is coming up. It's actually Friday, March 15th. That's this upcoming Friday. So please get your abstract submitted if you're planning to present and attend at that meeting. Additionally, I wanted to highlight the next North American section meeting, which is going to be taking place in Minneapolis, Minnesota in 2026. As you can see, the meeting will be taking place at the end of May, and it's going to be co-chaired by Tim O'Connell, who's actually a member of our Mid-Career Investigator Committee, as well as Jaap van Berlo and Julia Liu. And importantly, the MCI committee actually sponsors two great awards at this meeting. We have the Mid-Career Investigator Research Scholarship Award, which recognizes outstanding original research in the field of cardiovascular science by mid-career investigators, where we actually have 3 finalists that we shortlist to present at the meeting in Minneapolis. And we also have the MCI Leadership Award, which recognizes an outstanding mid-career scientist who's making major and independent contributions to the advancement of cardiovascular science, and also leading a growing research program that's likely to play a significant role in the future. The awardee will present a lecture at the Minneapolis meeting and also receive an honorarium and be featured in JMCC. So you can actually self-nominate for the Research Scholarship Award, but you have to be nominated for the MCI Leadership Award. So that's all from me. So I am going to turn it over to Eric, who will now introduce our first speaker.
Thanks, Mark. And thank you to JMCC for continuing to work with the MCIs, and thanks to the MCIs for putting on this great webinar series and allowing us ECIs to be involved. It's my pleasure to introduce Dr. Onur Kanisicak. He was a rising star early career investigator in his own right, and now is having a very productive lab as an associate professor. His background is with a significant expertise in cardiac and skeletal muscle stem cell biology. And more recently, his work has taken him into the cardiac fibroblast field that he'll talk about today. Originally from Turkey, he came to the United States where he did his PhD in Connecticut under the advisorship of Dr. Goldhammer. While he was there, he was studying renewable capacity of skeletal muscle stem cells. And then he moved to the Midwest, where he went to Cincinnati Children's for a postdoctoral fellowship under Jeff Molkenton, who we all know, and he's had a major productive career and has been an excellent mentor in the field. There, Onur studied c-kit stem cells and myocardial disease, and that seemed to pique his interest in cardiac fibroblast biology. He's had a remarkable career this far. When he first started his lab at the University of Cincinnati, having an excellent track record of mentorship in his own right, having several postdoctoral fellows, one of which at least has since gone on to start his own lab, and as a tenure line assistant professor. He has now moved to the University, Ohio State, the Ohio State University, where he's in the Department of Emergency Medicine and the Division of Basic and Translational Research. We're really looking forward to his talk today. Onur, it's a pleasure to introduce you, and please take it away.
Thank you, Eric, for this very generous introduction. I don't know if I deserve any of that, but again, thank you for inviting me for this very delighted webinar. And I'm going to try to go through as much of the data today to kind of justify the title I put it here. And hopefully we'll go through all these slides. I will fly through the published data. So most of it is from my postdoctoral years, just to give the background. But in a nutshell, hopefully I'll prove to you that cardiac fibroblasts or fibroblasts in general are important for both disease and wellness. Can everyone see me and hear me, hopefully? Let me see if I can move forward with the slides. All right, here we go. Just a quick introduction. Many years ago, back in 70s or 80s, fibroblasts have been described to what they do and what they're part of. They have been described as extracellular matrix producing cells. And since then, a lot of people actually use these cells in culture setting to discover many different biologies because they're very plastic and they have most of the capabilities of many cell types in our body. And back in the day, they still did not know actually as much as they can, find they are in culture, the identity, the origin, developmental origin, and the function of these cells to the extent that we can utilize their biology. Fast forward to now, and I keep putting this slide every time, and we're guilty of it too. What do we know about these cells? We actually still don't know exact identity, exact origin and function of these cells, but we were able to name multiple different, names for these same cells, based on our publication, our favorite name for them. In a nutshell, though, we agree a few things in the literature that these cells reside in the connective tissue of each organ. They're the stromal cells, and they're basically characterized as a non-vasculature, non-epithelial, and non-inflammatory component of each organ. There are many properties given to them or function associated with them, but essentially they support the function of the organ as well as the parenchymal cells. And biggest component of this function comes from extracellular matrix maintenance. And other characteristics, both in vitro and in vivo, that we know, if there's an injury or insult or stress to the organ or in culture, if you activate these fibroblasts, they actually become this interesting phenotypical change into a myofibroblast. And the reason we call them myo is like muscle-like. They form these stress fibers internally that allows them to contract, and that's what we call the myofibroblasts. And it's associated with increased extracellular matrix protein production. Now, there's a lot of debate here. Again, I have a lot of slides to go through, whether these two components of the myofibroblast or fibroblast activity is the same cell or separate. We're not going to get into that today, but if you have any questions at the end, we can talk. Then, this increases ECM. Is it a good thing? Is it a bad thing? So are myofibroblasts good or bad? Here's an example in a skeletal muscle setting where the skeletal muscle is regenerative, and you're just going through H&E staining here. This is how the normal muscle looks like. And after a massive degeneration, either with toxins or physical injury, the whole muscle degenerates and then regenerates. And during this process, it's known that these myofibroblasts produce the ECM to stabilize the organ so it doesn't completely rip off. And this happens to most of us, and we regenerate in a few days. So in that setting, fibroblasts, or myofibroblasts rather, are a good thing. But if you think about a chronic injury, constant injury to the muscle, or in a disease model like this Duchenne muscular dystrophy, where muscle constantly injures, these cells become activated all the time, and chronically they accumulate this extracellular matrix and make it fibrotic environment that actually leads to dysfunction of the organ. In the heart setting, it's even more drastic, not just for myocardial infarction, where you have an injury to the myocardium that leads to complete depletion of the ventricle, and then filled with this extracellular matrix. This is just time course of an injury in a mouse model. This is not necessarily exactly replicate of human, but in a nutshell, you have this left ventricle where you suture, cut the blood supply to the ventricle, and the ventricle muscle cells start dying. And in response, cardiac fibroblasts activate and fill it with excessive or protective physiological scarring, which is shown here with the pink for the collagen staining. And at the end, this heart does not rupture due to this physiological response. But then that leads to chronic strain and continuous expansion of the scar. So it's understanding of the biology of these cells is very important. Back to what I said before, the knowledge gap back in the day, or even today, includes the origin of these cells. Where do these tissue-resident fibroblasts come from, or fibroblasts, or rather disease-associated myofibroblasts come from? A lot of literature, if you look into, shows local and remote sources of endothelial, epithelial, and bone marrow-derived cells transitioning to myofibroblasts. But at the time when I started my postdoc, there was this one cell type, which is tissue-resident fibroblasts of the heart, was less studies due to its inability to study with markers and all that. So if we could understand the identity, origin, and function of these better, we can actually develop treatments and understand what they do in disease process, and can we actually identify mechanisms that we can revert the scar resolution, or resolve the scar that is produced, or stop it for heart failure. And that was the general idea that got my interest. Back again, just a quick background to this. The fibroblasts, resident cardiac fibroblasts, are again described as non-vasculature CD31 negative and non-inflammatory CD45 negative. But the other markers that will be positively marked them were very ambiguous. The vimentin, PDGFR alpha, are also labeling other cell types in the tissue. Collagens, they're expressed at baseline and activation, so they weren't really good, and they're extracellular proteins, so you can't really mark the cell. So the TCF21 arised from this one marker, seemingly a good marker at the time, and I'll talk about it a little bit. But more importantly, we're looking at this disease-associated myofibroblasts, and in that setting, the markers are even more ambiguous. The one thing that is defined in cell culture, at least, and in some disease, is this alpha SMA stress fiber formation. As you can see here on this large cell, these long processes are the stress fibers that allow them to contract. But other than that, ECM proteins, they're already there at baseline, they're increased, that's it. And all these markers you see were already there. But along the way, when I joined the lab, and the literature has been picking up on this one protein called periostin. And this is an extracellular matrix protein. It's not structural. It's very hard to describe, and I can have another hour of description of what this does and what I think it's doing. And we have studies just looking at the periostin protein itself as a ligand and as a glue to the whole ECM. However, in a nutshell, what I want you to do, understand for this talk is that it is only expressed during developmental stages, and in baseline uninjured tissues in our organs, the presence of this protein is undetectable. But after an injury, and here you can see that this is a myocardial infarction heart, and in whitish yellow, periosteum is labeled with immunofluorescence. You can see that it's right in the scar areas. So after injury, it's produced, but because it's secreted out, you couldn't really mark the cells that are producing this with this type of system. Therefore, we generated a Cre-LoxP approach where we put the Cre recombinase, tamoxifen-inducible Cre recombinase, at the periosteum genetic locus so that the Cre expression is faithful to the periosteum expression. So whenever it's periosteum expression, you got Cre. And if you put the tamoxifen, then you can modify another allele, which is in this case, the GFP reporter in its normal state. There's no expression of GFP. But if the cell expresses periosteum and we put the tamoxifen, it will turn GFP. And with this way, we hope to find which cell is producing this and study it. And again, I'm going to go through the data very fast just to give you a flavor of what we have done and what we think is happening. This is just demonstrating that we were successfully with tamoxifen dependency and with injury dependency, labeled the cells in the scarring area with green. And this is just showing you the same zone. And we were able to hopefully label the periosteum expressing cells. And we further looked at not only the heart, but every other tissue. Is there any expression of this at uninjured state? And looking at skeletal muscle, kidney, lung, liver, and skin, you don't really see much of these cells appearing. There's probably a turnover of it or a little bit leakiness of it, but really negligible in terms of what we label. However, in all these other organs as well, skin, skeletal muscle, and lung are the few I'm showing you here. After injury, you get a lot of these cells accumulating. So it seems like this process of periosteum expression after injury applicable to all organs, and that's probably why I'm going to retire from this. We're using this mouse. Quickly, we did a lot of interrogation of what these cells are. At the time, we didn't even know if they're fibroblasts or not. But what we did is generic marker studies, both positive and negative, and find out that they have a typical myofibroblast gene expression with alpha SMA, PDGFR alpha, and vimentin. Further, to both understand the heterogeneity within this population, if they exist, and what else are they contributing, we did a single-cell RNA approach to this and captured the GFP-positive periosteum expressed cells from MI, from uninjured tissue with the TCF21, that I'm not showing all the data, and kind of looked at this single-cell RNA transcription profile of it, and they matched the profile of myofibroblasts. The baseline resident, fibroblasts, very low in myofibroblast genes, but after activation, they become this myofibroblast profile. We also compared to the TCF21 lineage trace cells. Again, that's all published. You can look into that. And we did a lot of studies to find out the origin of these periosteum expressing cells are actually the resident TCF21 cells. And then we asked, okay, all these ECM and myofibroblast genes that you're producing, what is the consequence if you actually kill these cells or ablate them from the tissue? In the heart, it's a bit tricky, but we used the diphtheria toxin allele instead of a GFP allele. So in this case, instead of turning to a fluorescent expressing cell type, you actually die because of the diphtheria toxin, stops all the translation. So with this ablation approach, we did the same thing with MI. And as you may expect, in the first stages of MI injury, if you don't have fibroblasts producing the ECM to protect it, actually the heart ruptures, and that's what we saw. But if you look at the few that survived and look at the collagen expression, we actually saw a drastic reduction of the collagen in the scarring area. We have shown this also with different elegant ways of knocking out genes, and others also did, like SMAD pathway, TGF-beta pathway, and find out that these guys are the predominant fibroblasts or myofibroblasts produce the extracellular matrix. And that's what we found. And the origin is also in the published data. The real question is, what happens to these cells after this physiological, in myocardial infarction, for example, physiological scarring stage? What happens to these cells? Or in other diseases where you don't have this massive cardiomyocyte loss and you just get a little bit fibrotic response, what happens to these cells in the long-term chronic or when the injury may resolve, like I showed you in skeletal muscle? And those were generally what our interest has been in my lab. And as I am showing you here on a myocardial infarction model, you really cannot study what happens to them after resolution because there's no resolution. This whole ventricle stress actually expands to the septum and the right ventricle. And when you label these cells, you can see that they take over the whole heart and the heart fails. So you can't really study what happens to them if it resolves. Therefore, we start looking at skeletal muscle and skeletal muscle, because it regenerates. We thought that we might see a biological response that we're thinking. And in an injury model where you do cardiotoxin injection and the muscle regenerates, we looked at the acute stages of injury. Again, the green cells represent the periosteum labeled cells. They're activated, producing ECM. But when you stop it and you don't give tamoxifen anymore and the muscle regenerates, you can actually find these cells still present in a lower numbers in the regenerated areas of the muscle. And you can tell that by center nucleation and all other things that I'm not going to get into the skeletal muscle biology we're looking into. But this showed us there's a potential that all these activated fibroblasts at the time of injury might be still there after the resolution. Of course, the interest was a little bit in the heart to really understand what we can do there. So we introduced a new model that we think is a reversible fibrotic model or fibroblast activation model, where you give angiotensin and phenylephrine combination with osmotic pump for the 2 weeks duration. And during this time, you give the tamoxifen. So any cell, any fibroblast that activates and become periosteum will be labeled at this time. And then we rest at the heart because the pump depletes, the angiotensin goes away. This doesn't create a lot of cardiomyocyte death. So then we hope that the heart will recover. And what I'm showing you here is, again, similar to the skeletal muscle, the GFP component. At the 2 weeks of angiotensin, the peak injury stage, we have a ton of these lesions of fibrosis and fibroblast accumulation, both in whole mount imaging and histology. But if you were to wait only 2 weeks even, you start seeing a massive reduction of these cells. But obviously, we wanted to wait longer because what if the clearing of these activated or previously activated cells takes some time? So we waited up to like almost 8 months now, and they never really completely disappear. And this gave us the idea that potentially there's an activation and deactivation happening for these cells. A bigger question we also asked related to the fibrosis, do the fibrosis goes away too, or is it permanent damage? And what we show here with the collagen staining, that at the time of peak of stress, we have fibrotic lesions, but it goes away, dissipates. Cells remain, fibrosis goes away. We looked at all the different type of cells and all these different physiologies and echocardiographic methodologies. And what we found that a lot of the cells that responds to this injury state actually revert back to their original numbers. And maybe there's a little bit increased number of fibroblasts residing in these hearts. But for the most part, they go back to normal. And a heart a little bit hypertrophies, but go back to normal. So the questions become, what happens to these cells? These quiescent fibroblasts activated, became periosteum. What happens? And you can see from the cheat sheet here, the deactivating and dedifferentiation actually happens. We looked at the common markers, myofibroblasts, alpha SMA expression in these green cells. That dissipates, and you have green cells that are not alpha SMA anymore. We look at the periosteum expression around these cells. That dissipates. But the PDGFR alpha, which is a marker we use predominantly for labeling both activated and quiescent fibroblasts, they remain to be positive for these. So that's interesting. Then what we did is we went to each of these critical time points at the time of injury, at the acute reversing stage, the 2 weeks after resolution, and then a later stage, resting resolution stages, and did another single cell RNA sequencing approach. And what I want you to take away from this, each of these lines are single cell gene expression profile. Dark orange is higher expression, and black is low expression or none. What I want you to focus here is this mid belly here. What I'm showing here, the myofibroblast genes. And on these quadrant, on this left, these are more quiescent cells. And this is over here, activated periosteum positive cells. As you can see, they're highly expressing myofibroblast genes. And then during the deactivating stage, they reduce those. And once they become more de-differentiated or back to resident state, which they don't actually, they similarly show the same transcriptomic profile as the resident cells. But if you focus a little bit and squint your eyes, and you might just see this group of genes I'm showing all the way on the top and all the way in the bottom. As you can see, some of these genes that were not, normal expression goes up and stays up at this deactivating stage and go back to normal. And reversely, some of these genes that were there already and upregulated completely shuts down in this deactivating state and then come back to a more normal homeostatic level. And looking at these, sets of genes, which we thought that could be interesting to look at, we found, okay, before that, let me give you what's happening in here. As you can expect, that the cell cycle genes and myofibroblast genes went up during activation and went down during deactivation. But back to those two interesting positions that we thought, what if those are the genes responsible for reversing the fibrosis that is accumulated? And that's what we thought, that this area, this type of cell type, is a special state for these fibroblasts. Looking at the main things that we were thinking is the MMPs, the enzymes that degrade the ECM, and the THIMs that actually inhibit those MMPs. I'm showing you two slides. One where resting fibroblasts and myofibroblasts are compared, and periostin is your anchor for activation state. As you can see, a lot of the MMPs did not necessarily change their expression, but some increased specifically in these cells. But the THIM1, the inhibitor for these, is skyrocketed. So in that balance, the MMPs that are produced in higher numbers could not degrade the ECM because the THIM is there. Or another hypothesis that there's a higher amount of metabolism happening, both degrading and building the ECM. And as you can remember, these produce a lot number of the ECM proteins, so that makes sense. But if you look at this, and I'm hinting here what I want to call them, the fibroclast, which is the ECM degrading fibroblast state, which is that 2 weeks deactivating state, compared to the myofibroblast, what happens is MMP expressions kind of stay similar, except some of them, but the THIM expression plummets down. So it seems like the cell that activates, when they need to build the ECM, they produce the both MMPs and the ECM and the inhibitors for the MMPs. But once the environment is going back to normal and injury signals go down, what they respond is they don't completely go away and shut down. They actually change the balance of building the ECM to allowing to degrade. And one of the experiments, again, we visited a cell ablation approach to this. If this is true, and the ECM that is built has to be taken away by the same fibroblasts, we hypothesize what happens if we kill them at the time that they're going to do that. Just a quick understanding here. We used the whole fibroblast Cree system here and ablated all the fibroblasts only during this deactivating stage, not during activation, but only during the deactivation. And what we saw there is that this is a control that is activated and deactivated heart, looking at the series, picoseries staining with the red showing the collagen depositions. There's some remaining. These hearts are never really at baseline anymore after that insult, but pretty much is cleared out. But if you ablate these fibroblasts, then that picoseries red staining remains in higher amounts, and fibrosis is not depleted. So showing us that the cells that are responsible to both build that ECM are also required to degrade the ECM. And that's the balance where, fibroblasts are known, or seemingly both good and bad, but it's more about the injury and the organ, the regenerative capacity of the organ. Now, I'm just telling you the fibroblast standpoint of it, but I want to also emphasize that they don't do it alone. A lot of the MMPs and the THIMs and all the enzymes actually actively dissolve the fibrosis also comes from the macrophages and their states, and we're studying them, but that's not the whole point here. And so I wanted to emphasize for this part of the talk, and I don't know how much time I have, Eric, maybe 5 more minutes, if I have. I wanted to say that this approach to resolving fibrosis has to come from a multicellular approach the damage signals coming from here, and all these other cells around it. So if I may, I'm just going to give you a quick preview of a paper we just submitted, and it's out there in bioRxiv if you want to read it. Goes into that sentinel side of things a bit, and I'll finish it with this. Many studies, as well as ours, actually start seeing this diversity in the fibroblast populations, calling them either fibrotic or matrix producing or myofibroblasts or cytokine producing. So for that matter, we looked into whether can we take these myofibroblasts and with insult and with artificially, in my opinion, in that sense, push them to be more immune modulating phenotype. And with a collaboration, and I'm going to just, again, this is published, a collaboration with another group, what we did is we isolated resident bona fide fibroblasts and then put the interleukin-1 beta induction on them and show that those fibroblasts that are meant to be producing a fibrotic response actually can be influenced to become more myeloid-like cell and produce a lot of cytokines and play a role in inflammation in the heart. But in the heart, we do not have a massive amount of infection or need for this flavor of fibroblasts. And it seems like this flavor of the heart is about 3% or so. So we went to another organ where we know that it's going to be exposed to a lot of pathogens, which is the lung. And we chose a pathogen that will not necessarily cause disease in an immune competent background, but it's a massively important health hazard these days, Aspergillus fumigatus fungi. If you're healthy, if you're immune competent, you get a little bit phlegmy when you rake your leaves in the winter, but you don't get much disease duration. But if you're immune compromised, despite all the antifungi and antipathogen and drugs they give you, there's an almost 90% lethality of this. So if you're organ recipient or immune suppressed individual, this is a big hazard. But the question we were asking here in this setup is that the field thought that the macrophages in the alveolar space is responsible to clearing these pathogen spores. And our question was, what are the fibroblasts doing in that setup? If there is a true cytokine and crosstalk between the fibroblasts and the myeloid cells, instead of the heart, we can actually study it here. And I'm going to finish up with 2-3 slides and we'll be done. And what I'm quickly showing you here is similar to the MI model and all the other injury models we have done in the heart. The dependency of labeling of the activated fibroblast depends on tamoxifen and depends on the pathogen being there. But surprisingly here, what I'm trying to show you and emphasize is that this is an immune competent host that should not have any disease or this should be an asymptomatic challenge to the lungs. But what you can see here, that a lot of the fibroblasts in this quote unquote healthy mice activated. And we thought that this activation is actually causing that underlying, fibrotic response, the acute response. And when we were doing these studies, COVID hit, and now we know that lung fibrosis and in response to pathogens is a big deal. So finishing up with this, what we asked a similar question in the heart: are these activated fibroblasts in response to pathogen critical for immune regulation? One interesting thing we saw is the cross anatomical congregation of these activated fibroblasts to the myeloid populations that are activating. Again, this is submitted. It's in the bioRxiv. We have a lot of data showing this. But the real question we ask is that if we take these fibroblasts that are activated, ask the question, do they produce any inflammatory cytokines? We see a massive activation of these. And then the natural question became, again, is what happens if they're not there? We know in the immune suppressed host, if we give the aspergillus, they die. And this black line over here shows you a normal animal dying in about 7 to 10 days after this infection. But when we ablated the fibroblasts within this environment, we saw rapid decline of health, and they completely die fast. And if you look at the fibroablated lungs, you see this blood accumulation, and further investigation show that we have a lot of hemorrhage due to the ablation of these fibroblasts and a lot of angio invasion of this black little hyphae, the germinated fungi going into the blood vessels and dissipating in the whole animal and causing death. So we think that fibroblasts are not just these ECM regulating, scaffold building, cell type, but it's really actively involved both protecting the organ by scarring, physiological, or be a sentinel and crosstalk communication to synchronize the response of other cells. And these are some of the interests we're trying to figure out, whether they directly find it or not. And I'm going to stop it there and thank all the people actually did the work. The one good thing about mid-career stage is that, or actually having a faculty position, you don't have to do the experiments anymore, which sucks because they find the innovation and you just put together a talk. But these are the people and many others that are unnamed here helped build this career that I'm immensely enjoying. And thank you for your time.
Onur, that was an amazing talk. Thank you for taking this all through the historical perspective of your work through your postdoc and the stuff you guys are doing now, which is really exciting. As Mark has posted in the chat, please, if there are questions, submit them to the Q&A. I can spend the next hour asking him questions that he's going on, so maybe I'll try at the next conference to grab coffee. In the meantime, I do have a couple, so I'll kind of just start it off a little bit. I came across a paper from Amy Bradshaw's group a year or two ago that had a similar type question as you were. They used the TAC model instead of the ANGV model, and they did this really impressive maneuver where they did TAC and unTAC, where they actually removed the suture, and they did not go down to the depth of the mechanism that you have, but they saw it, of course, later time points of pressure overload. There gets to be a period where you can't revert, you can't regress back. So in the context of the, I forget what you called them, the ECM degrader fibroblasts, what do you think's going on there? Are they dying off? Are they becoming senescent? And now
Right.
one expression is no longer able to do what it's doing? Or do you have any insight?
Yes. I'm of course showing you the parts that makes a good talk, but we're actually looking at the senescence aspect and what happens to the cells that die. Is there a difference between the cells dying or the cells remaining? And what I'm not showing here is actually there's a de novo activation of fibroblasts during that reversal stage, which we think, it's a fight between or synchrony between deactivating cells and newly activated cells to degrade the ECM along with macrophages. But back to the question, this injury, if you want to study heart, which I do, is very, hard to do in some models where you induce cardiomyocyte death. So we have mice, they're mixed backgrounds, so their response to angiotensin is mixed a bit. And as you know, angiotensin actually causes AAA, so males are more susceptible sometimes. And if you push them too far, they don't come back. So that lesion that I showed you at 2 weeks, if that lesion expands too much and restrict the cardiomyocyte health and they start dropping out, that heart doesn't come back. So we have an exclusion, way to say that, okay, these hearts, we cannot study reversal. We only study on the ones that reversed. TAC on TAC, we also did, and back in the day. Honestly, that's a bit more challenging when it comes to injury, surgery part. So we have better survival of angiotensin phenylephrine. And so we have done that, but we have done micro MIs, TAC on TAC, and we saw similar phenomenon. But I think there's other studies like Taban Hale, who has done hypertension model in rats, and she sees similar but not exactly response from these cells. She does see the reversal of the hypertrophy, but one thing we disagree is that, which I didn't show you, if these hearts that are reverted carrying previously activated fibroblasts, they were challenged again, the response is much worse. The previously activated fibroblasts actually expand much more. They're hyperactive. We have actually in vitro cell culture approach to this. that they massively activate again. So they remember, there's a memory of the activation. And we think that's detrimental and the key to the chronic injuries that we talk about in other organs that are regenerative. But she thinks that actually they don't respond as much because they are now desynthesized. So that's what we study. I don't know if I answered the question or answered 5 other questions that you may have.
That was perfect. And we have some questions in the chat, so I'll get to those with just like a food for thought that I'd be curious now that as patients are taking semaglutide, tirzepatide for fibrosis associated with cardiometabolic disease, if they come off that regimen, then they see what you're seeing, that they're more predisposed to a hyperreactive state. But we'll get to these Q&A questions. So we have one from Liming Pei, and is that possible that both pro and anti-inflammatory, or M1, M2 as some preferred fibroblasts, in different organ disease stage context?
Yes. Answer is yes. We do see the diversity, and I think I'm not sure if they're referring to macrophages as M1, M2, but we also think that there is a quote unquote F1, F2, fibroblast type. Yes, there are pro and anti inflammatory cytokine-rich states of these cells. In fact, if you look at the early stages of MI, fibroblasts are pro-inflammatory producing. Only at that reversal stage we see anti-inflammatory, or rather, cytokines that probably polarizes the macrophages to more M2. I'm just making this up right now. We don't have evidence, but someone is working, Daniel is working in the lab for this. So I think they're influencing the polarization of myeloid cells as well as infiltration.
Excellent, thank you. And she had a follow-up question about one dominating over the other that you just answered, so thanks. The next one is from Andrew Gibb. He says, great talk, Onur. How do you think these cardiac fibroblasts coordinate/integrate the barrage of competing signals to regulate their potential fate? And he says, that is, how does a fibroblast know to become inflammatory because IL-1 beta signaling versus profibrotic via TGF-beta signaling?
Right, beautiful. Here's my hypothesis, and we're studying this, so don't scoop me. So we think the early stages, particularly with MI, early stages have a lot of cardiomyocyte dropout, right? There's a lot of damage signals going around, as well as platelet cells coming in, bringing massive amounts of TGF-beta. In that environment, the response, just like you put TGF-beta in the culture to a fibroblast, becomes profibrotic, right? Myofibroblasts produce ECMs. But the nuance is, after that, if you study a regenerative system like skeletal muscle or angiotensin that we're using, there's no damage signals. And there is a normalization of the cardiomyocyte function, because the angiotensin, at least, not just activates the fibroblast, but also triggers the cardiomyocytes to beat more, and vasculature stress and all that. Once those dissipate, the crosstalk between those cells to fibroblasts dissipates. And then they can sense that, okay, we don't have to produce as much. And in vitro studies that we're actually conducting right now, utilizing different matrices and stiffnesses, we know that the fibroblasts actually also sense that. So once the combination of all these, both cytokine and environmental senses, periostin is one of them, and that's the other paper we're working on. They reduce that phenotype. Once they reduce it, that's a positive feedback, and they revert back to normal.
Perfect, thank you. That was wonderful information. Those are all the questions and great timing. I think with that, I'll turn it over to my colleague, Dr. Gary.
All right. Well, it is my absolute pleasure to be here and also to introduce Dr. Emily Tsai, who is the Florence Irving Associate Professor of Medicine at Columbia University. She's a physician scientist. She is a cardiologist and advanced heart failure transplant specialist, and does it all. She graduated with honors from Harvard College and Harvard Medical School. She completed her internal medicine training at UPenn, and then cardiology fellowship and advanced heart failure fellowship at Johns Hopkins. She completed her postdoctoral training in the laboratory of David Cass, who we all know and love, and focused on cyclic GMP and nitric oxide signaling there. in pathophysiological cardiac stress settings. In 2009, she established her laboratory at Temple University, and then in 2015 joined the faculty as associate professor at Columbia University, where she works on molecular and integrative cardiac pathophysiology. So she and her laboratory study specifically nitric oxide signaling in the heart, RV dysfunction, and cardiac complications of COVID-19. And most recently has worked on sex differences in RV remodeling, which we are going to hear a lot about today. She's received many, many prestigious awards, too many to mention, including the Presidential Career Development Award and the J. Cohn New Investigator Award. She has been a mentor to numerous grad students, cardiology fellows, postdoctoral fellows, and it is really just a pleasure to have her here today. So take it away, and thank you very much, Dr. Tsai.
Thanks so much for the invitation. And I've got to say, I'm not doing it all. I often feel like I'm just, barely surviving. And excuse my voice, I caught a cold. Okay, let me make sure I can share my screen properly after all these years of Zoom somehow. Sometimes still have some technical challenges. Okay, let's see, how will I do this? Sorry, oops. I think I'm just about to do this correctly. Sorry. Okay. So, do you see some? Oops.
Looks great.
Do you see a slide?
Yep, you are good.
Okay, great. Because it looks like there are all these other windows open. So, so yeah, thanks for that introduction. And this is a relatively the most recent sort of area of focus that my labs kind of entered into. So what I thought I would do is kind of present a little bit more of sort of a clinical introduction, why the RV, just sort of a little bit of background. I am an advanced heart failure transplant cardiologist, and sort of in my clinical work have encountered a lot of RV failure, and very little is understood about sort of the remodeling of the RV at the sort of molecular level. And that to me became a very interesting topic. And so we've published in that area, and this is relatively new. So don't have any disclosures. As an overview, just thought I'd kind of divide this into 3 sections. First, what is RV dysfunction and failure? Why does it matter? And how does this differ from the LV in terms of remodeling? Secondly, what is sexual dimorphism of RV remodeling? What are the differences between males and females, and what underlies that? And then sort of lastly, kind of more of an overview, sort of what we've learned thus far about the mechanisms of sex differences and pressure overload RV remodeling. And actually, I have lots of questions for Onur because we have sort of gotten into some about some fibrosis and sex differences in fibrotic remodeling, which has not been my background, but sort of the researcher, the scientist who kind of led that is very interested in fibrosis. So let's see now. All right. Oops. So, just starting with the first section, what is RV dysfunction and failure? So, much like LV dysfunction, it refers to impaired RV contractility due to hemodynamic stress, injury, myocardial dysfunction, sort of innate myocardial dysfunction. And basically you can think of it in terms of like 3 sort of groups, sort of hemodynamic groups: pressure overload, volume overload, and contractile dysfunction. And essentially much of RV dysfunction and failure is manifests in terms of symptoms that reflect elevated central venous pressures. So sort of unlike LV failure, you see some sort of GI manifestations of nausea, early satiety meaning like just getting full very easily or being able to only eat like little bits of food at a time, loss of appetite, and then sort of it also hepatomegaly, ascites and edema, and then of course fatigue and poor exercise tolerance. So that's sort of the clinical sort of picture, symptomatic picture of RV failure. And then in the most sort of advanced stages, you have sort of multi-organ dysfunction. Certainly there's increased arrhythmogenic potential, so lots of arrhythmias, but importantly, sort of kidney failure, liver failure, and sort of as a reflection of just all the sort of GI failure is cachexia. Now, what causes RV dysfunction and RV failure? So it's not just sort of the cardiac causes that we think about regarding LV failure, but it also involves pulmonary or includes pulmonary etiologies. So we've got the usual suspects of MI and cardiomyopathies, valvular disease that lead to left heart failure. And with left heart failure, whether you've got a reduced or preserved ejection fraction, you ultimately get an increase in the LV filling pressures. And so then that kind of, that sort of like backward increased pressures can lead to what we typically call post-capillary pulmonary hypertension. And in very severe cases, there's also chronic vascular remodeling, and it becomes an even more severe degree of pulmonary hypertension. And so that increase in pressure overload leads to the RV dysfunction. And then you can also imagine the things that would increase RV pressure overload would also include lung diseases. So pretty much every lung pathology can lead to pulmonary vascular remodeling and then pulmonary hypertension. Sort of maybe what most people think about and what a lot of the sort of historical research has been on is pulmonary arterial hypertension, which is actually a rare disease, but you can imagine it's sort of more pure in that it's just RV afterload increases and leading to RV dysfunction. So, why does this matter and why am I interested in RV dysfunction? So, what this kind of hints at when I talk about, when I mentioned right heart, or rather reduced LVEF and preserved LVEF, is the fact that RV dysfunction is highly prevalent and it portends poor outcomes. So just to show sort of these pie graphs, their RV dysfunction affects in various studies 60 to 76% of patients with HFrEF, but also like 30 to 50% of patients with HFpEF. And as LVEF decreases, there's an increased prevalence of RV dysfunction. And then regardless of LVEF, there have been several imaging studies, MRI, by echo, looking at heart failure patients. And they've shown in these different modalities that a reduced LV ejection fraction, or RVEF, leads to poor clinical outcomes, worse clinical outcomes. So event-free survival drops, and that's been consistently shown. And that's also the case in sort of the pulmonary vascular diseases. Really what kills those patients is RV failure and the consequences of RV failure. And so why does that still happen? We've got these great medical therapies for heart failure, right? So many, especially recent trials that have added new pharmacologic categories to what we call our guideline-directed medical therapies. And they've been proven by multiple large clinical trials to be beneficial to heart failure patients, whether they improve survival rates, either overall or cardiovascular survival, or reduce heart failure hospitalizations. And so, we've got what we call the 4 pillars of GDMT: beta blockers, mineralocorticoid receptor antagonists, SGLT2 inhibitors, and then what's usually grouped together is ACE inhibitors, ARBs, and angiotensin receptor blockers, and ARNI, so the angiotensin and neprilysin inhibitors. So those are like, the 4 big pillars, we call them, that those are the meds that we want to optimize our patients on. But, when you look at, what's been found in trials of HFpEF or anything that's looked at RV dysfunction, and most of that has been in pulmonary arterial hypertension clinical trials, they haven't proven to have, great survival benefit or clinical benefit at all, or they just haven't been studied. So why would there be a difference in sort of how the RV responds to the LV? Responds to these medications. So, the RV fundamentally differs from the LV. Sort of embryologically, the RV is derived from the second heart field, whereas the LV is derived from the first heart field. And, it also has several differences in terms of its sort of function, morphology, and physiology in response to stress. So the RV faces a low resistance circulatory system, the pulmonary vasculature, which is also low pressure. The LV, in contrast, works against a high resistance, high pressure vascular system. The RV has very thin walls. The LV has much thicker walls. The RV contracts in a peristaltic-like motion from its inflow to outflow tracts, whereas the LV, contracts in a radial thickening and contraction pattern. And because of these, they have very different pressure to volume relationships, and their responses to increasing pressures are different. So like the RV, although it can handle volume overload quite well, doesn't do so well with pressure overload. Its function drops precipitously, whereas the LV can sort of maintain its function better. And what's been sort of found so far is that there truly are signaling responses that differ between RV failure and LV failure. There are things that are similar, but metabolic adaptation involved, oxidative stress is involved. You've got EP remodeling, angiogenesis, and on. But there are responses that are quite specific or significant in RV dysfunction, and so while these are kind of the sort of highlighted differences, what we know about the molecular signaling is actually like pretty broad. Like the details of those mechanisms aren't well known. Like whereas, sort of we think about the intracellular signaling in cardiomyocyte hypertrophy failure of the LV. We've got like so many different really busy signaling signaling cascades that like you could pull up a review and it would just be like 100,000, not exactly, but, or maybe probably, we just haven't found all those 100,000 sort of signaling partners, but the RV is not as well understood. And so, that's sort of why I become so interested in RV pathological modeling. Now, what is the sexual dimorphism? So what was noticed first in the pulmonary arterial hypertension population was that even though PAH was more prevalent in women, the women actually did better than the men. They had better RV function, they lived longer. And so, other than the fact that women in general are maybe the advantageous in some ways gender, what happens with respect to PH and RV remodeling is that the main concept is sex hormones have different effects on remodeling. So maladaptive refers to what's seen with testosterone mediated signaling in response to pressure overload, whereas women show an adaptive RV remodeling to pressure overload, and that's largely due to sort of the cardioprotective effects or actions of estrogen signaling. And so, sort of in general, testosterone signaling is associated with increased fibrosis, increased hypertrophy, and thereby worse RV function and a lower survival rate, as the survival curve shows. With estrogen, the cardioprotective actions include sort of a reduction of the inflammation, sort of reduction of oxidative stress. There's less mitochondrial dysfunction. There's less proapoptic signaling, and thereby better RV function and higher survival rates. So that's just like very in general. So what have we learned in our entrance into this field? So I'm going to talk about our sort of two projects, one that has been published and another that hasn't been published and is in revision. And the first one is really related to the structural remodeling. And this is where like some fibrosis or ECM remodeling comes into place. So I would love to, spam. I would love to get Onur's input. And then the role of soluble cyclase is, soluble guanylyl cyclase is sort of the central sort of focus of the second project, in large because as mentioned, I, in my postdoctoral work, had focused on SGC signaling. So, so my lab, you know, and trying to integrate the things I learned from Dave and sort of moving forward with, you know, more sort of cardiac myocyte biology, try to integrate all these techniques that, you know, incredible mentors have really exposed me to. And so here is kind of the experimental design of this particular paper, but it kind of follows sort of the you know, pattern that my lab has taken. So here in the study, we had male and female wild type mice that underwent either PAB, pulmonary arterial banding, think about TAC, but just on the pulmonary artery, versus sham. And we let them go, you know, for like 9 weeks post-surgery and did serial echoes at 3 and 6 and 9 weeks. And then at, you know, the endpoint, so study endpoint, we had, you know, subgroups that underwent different analyses of function and structure, right? Imaging, histology, invasive, you know, PV loops, RV PV loops. And I'm just showing, you know, some of the findings, some of the data, and you can, you know, get more details from the paper. But you know, in our model, we certainly replicate the sexual dimorphism that's seen in patients. So the post-PAB survival is poor, worse in the male mice compared to the female mice. And then sort of one parameter of RV function by echocardiogram is TAPSE, tricuspid annular, annulus planar systolic excursion. And you know, the higher that is, the better. The lower it is, you know, the worse the function. And while in sham mice, that function is, you know, equal, there's no gender difference. There is a difference in the PAB animals. So of course, they both have sort of some RV dysfunction compared to the sham controls, but they're significantly worse in the males. And that also holds true. And what we see actually is in terms of hypertrophy, using the Fulton's index, RV weight over the combined weight of the LV and interventricular septum, that you certainly see hypertrophy in both in response to PAB. And so looking at this on the histological level, sort of on WGA staining, you could see sort of the male mice, female mice at sham and PAB. You can see the cross-sectional areas of the myocytes all increase within the PAB mice compared to their respective sham. But it's also, you know, larger in the males than in the females, so not quite as much myocyte hypertrophy. And then in looking at fibrosis on Pegasius red staining, found was sort of the total collagen in terms of the percent of sort of the pink staining in relation to the area of the ventricle. There's an increase, there was an increase in the male PAB mice compared to sham, but not so much so in the females, and significantly less than here compared to male PAB. On top of that, I was really interested in looking at like the pattern of fibrosis or perivascular versus replacement fibrosis, and doing so, she actually found that this bar graph shows sort of the replacement fibrosis, animals that had mostly replacement fibrosis, and those that had, I guess, more perivascular fibrosis, or sort of the ratio of that for the animals. And you could see the male PAB had far more replacement fibrosis at 6 weeks, and that was persisted even as time progressed. And so in trying to kind of tease out what about it ECM remodeling is different in response to RV overload between the sexes, Laura looked at further sort of collagen degradation using a collagen hybridizing peptide 5 FAM conjugate. It's referred to as F-CHIP, and that allows her to assess quantitatively sort of the amount of RV collagen degradation. And it was very obviously much greater in the male PABs than in the females. You don't see as much collagen degradation. And then in looking at sort of collagen structure and cross-linking, while these images don't show as well, sort of the quantitative analysis shows there really isn't much difference in terms of cross-linking by this particular assay. And so without going through like every detail and finding of the study, basically the sort of summary conclusion of Ilaria's work was that the RV-ECM instability and chronic RV inflammation distinguished the male from female PAB. So I didn't show any of the the assessment of the inflammatory response, but if you looked at the amount of like activated macrophages in the RV of these different cohorts, and did a correlation with the degree of fibrosis, and we could see that there was certainly a high correlation of increased activated macrophages in the male maladapted RVs, and then there is she looked at different sort of matrix proteins, different cytokines that are pro-fibrotic, and kind of basically the bottom line was sort of this ECM remodeling, sort of the inflammofibrotic process, and that's sort of as far as we've gotten so far in terms of the mechanism. But it's certainly this highlights what Onur was saying about sort of the interaction and the communication between fibroblasts and immune cells. Okay, so then so our second project I wanted to present is regarding sort of the role of soluble guanylate cyclase in this. And I think one of the reasons I thought to consider SGC, or actually sort of the new nomenclature, is GC1. The reason I was interested in like GC1 as a potential mediator of these sex differences is, as Mark knows really well, estrogen regulates NO signaling, and GC1's a NO receptor. And actually, a postdoc that joined my lab who was particularly interested in sex differences and PKG signaling, and so I was very fortunate for him to join my lab and kind of help with leading this project. So what we did was with the collaboration of Andreas Fried from the University of Würzburg, created a cardiomyocyte-specific GC1 functional deficient mouse. So mice were flocked at exon 10 and crossed with the myosin heavy chain promoting Cree. And so in the progeny, we would feed both sort of the Cree negative and Cree positive with tamoxifen to kind of control for the effects of tamoxifen. So you'd have control and knockout to compare, male and female to compare, and sham and PAB to compare. So it's a lot of mice, but we were able to find that, okay, in terms of sort of the knockout mice, there are no differences in the expression of GC1 in male or female. And in fact, there was a decrease in its expression compared to control. We tested the function of these cardiomyocytes by basically stimulating them with an NO donor Dino and measuring baseline cyclic GMP compared to those that were stimulated with the Dino. And so yes, in terms of function, there's also marked, there is increased production of cGMP in response to Dino in the control mice, but not so in the female, or not so in the knockout mice. Although so although there seems to be there's a difference between control and knockout in both. Okay, so that's at baseline. And then in PAB animals, we should kind of look at function, the physiology. One of the studies you do is to look at the sort of exercise capacity, doing the sort of treadmill testing of the mice. And here you can see the PAB mice, the females controls actually do better than the males. But in the knockout and in the PAB animals, the sort of female advantage is abrogated. So the knockout mice that are banded do poorly, and the only difference between in the females is the cardiomyocyte sort of functional deficiency in GC1. And then here in the survival curves, I don't think this projects very well, but here the top line is female control mice versus female knockout, and on the right, same for the males. And the male mice did poorly. Didn't matter whether or not they had GC1 expression and function, they did equally poor. But there's a significant drop in survival rate in the myocyte GC1 knockouts in the females. And sort of trying to understand the functional differences, their structure function. We looked at the histologic remodeling of these animals and also found that there was sex-specific effects of myocyte GC1 deficiency in the histological remodeling of the pressure overloaded RV. So this time kind of expanding our assessment more, and largely because these three histologic features are characteristic of RV, the failing RV. And so that's hypertrophy, fibrosis, and capillary density. So capillary urea fraction is considered a basic classic characteristic of the failing RV. And sort of in the quantitation of this, what was striking is the effect of knocking out the GC1 in the myocytes and the females. They're the ones that showed the maladaptive or more maladaptive remodeling at an equal sort of severity as the males. And to try to kind of tease out, well, we know that myocyte GC1 signaling would impact hypertrophy. The roles are a little less known in fibrosis and in capillary sort of rarefaction or microangiogenesis. So we did like a Pearson's correlation between measures of RV function by echo. So here, fractional area change. We also looked at TAPSE. And here I'm just showing the data for capillary density and capillary cardiomyocyte ratio We did this as well for, myocyte cross-sectional area and percent fibrosis. And these are just all the mice, regardless of, their sex, genotype, and surgery, just to like purely like we're just looking at sort of RV function and these histologic findings. And these are our paired data. Like the RV fractional error shortening is for that specific, is the same animal as that the capillary myocyte ratio is being reported. And so we found very strong correlations between the two, and sort of essentially like the highest for these markers of microangiogenesis or capillary rarefaction than for fibrosis or hypertrophy. Kind of equal with hypertrophy, but sort of more interesting to us was this whole idea of the microvascular dynamics remodeling. And kind of to look at the estrogen role in sort of these processes, Nobu also did studies comparing females that had intact ovaries versus those who had underwent ovary ectomy prior to PAB. And sort of they essentially replicated, or again, like supported the fact that sort of it was sort of estrogen that that was sort of regulating the adaptive remodeling in the females, and that even with estrogen, without the myocyte GC1, you would lose the adaptive remodeling. And then he took this to sort of test in the in vitro setting, doing this endothelial cell migration assay, so co-culturing sort of myocytes that are isolated from our different cohorts, and then stimulating the co-cultures with estradiol and looking at sort of VEGF-A, both expression in the myocytes, and then looking at VEGF-A in the actual media from these co-cultures, and could show that yes, you need that the control animals, where is this? So that in the control animals, there was migration, and in response to estradiol, but in the knockout myocyte co-cultures, that was lost. There's a lot more that Nobu also analyzed in these animals, including doing PV loops and other molecular studies. But this is kind of the schematic of the summary of his studies, which is, okay, we've got sort of the male and female mice, control and knockouts that were subjected to pressure overload. And we saw, as would be expected, sort of the adaptive RV remodeling and the maladaptive RV remodeling in the females versus the males. And sort of most strikingly in terms of the microvasculature of the RV, whereby sort of the microvascular sort of capillary density and capillary to myocyte ratio is way better in the females than the males, and that advantage was abrogated with the loss of myocyte GC1. And that also correlated well with like RVPA coupling, which is sort of a measure of sort of work efficiency of the RV. And then kind of trying to put it together in a signaling aspect, that GC1 here is really necessary for the estrogen stimulated sort of pro-angiogenic signaling, so that sort of in the females we have sort of a pathway of anti-capillary rarefaction and sort of pro-microvascular angiogenesis, and that is one mechanism by which we think the sexual dimorphism occurs in RV pressure fluid remodeling. And so at this point, sort of the next steps are really sort of our interests in the remodeling of the microvasculature and sort of the crosstalk that occurs between sort of the myocytes and endothelial cells and smooth muscle cells. Maybe another RO1, but at my sort of stats so far, I think I'm going to need to get up to the plate a lot more times. So here's the so hopefully. So hopefully, you've got a general sense of the answers to these questions, understanding, what RV dysfunction and failure is, why that difference and how it differs from LV remodeling. Sort of, I did a very simplified version of, what underlies sexual dimorphism. It's really sort of the main framework or paradigm is related to sex hormone regulation, but there are also non-sex hormone mechanisms that have been hypothesized and have been supported by some data. And while we're kind of entering this area, there are a couple other groups that have been studying this for much longer and have elucidated very interesting effect of estrogen signaling on sort of the contractile function of the pressure overloaded RV Tim Lamb's group, who I think is now either in Colorado. I think he's in Colorado. In any case, I'm very lucky to have incredibly great lab members. Nobu led all the work on the GC1 deficient mice. Ilaria led the work on the sort of fibrotic remodeling differences, and Wendon and Christos have contributed those and have their own projects that they lead. There have been this sort of list of other sort of collaborators and just mentors is not complete here, and these are various funding agencies that have helped sort of my career along the way, and so I'm obviously incredibly grateful to them. This has been sort of a long process, and with that, I'll take any questions. I'm sorry, I'm not sure how much time I took, but hopefully I kept within the time limit.
Yes, that was fabulous. Thank you so much. Very intriguing talk. So I have several questions. I think one of the things that in looking at your GC1 deficiency model that comes to mind is how much of this do you think is the phenotype that you're seeing specific to RV mechanisms as opposed to, the mechanisms that we know about with sex differences in the LV, obviously with PAH, nitric oxide signaling is very important, but I'm wondering if you could just spell out some of those very unique aspects.
Yeah, so, there are sex differences in the in sort of LV dysfunction. There's like mixed data about that. And also it's not as, I guess, pronounced in the sense that those sex differences some of the sex differences are said to be due to sort of the specific type of cardiac injury in terms of how androgen signaling goes, and then in estrogen signaling. There's a lot that's unknown, but with sort of on the RV, whether it's sort of pulmonary vascular disease or heart failure, sort of the ultimate kind of stressor is increased pressure overload. No one knows if there's a difference in terms of sort of sex hormone receptor expression, like in the RV and LV. That hasn't been done. But sort of the, as I mentioned, in terms of like how the RV responds to stress and sort of the differences, all the things that are fundamentally different, that could be very well sort of what highlights its role in sort of RV remodeling than in LV remodeling. You know, because the RV also kind of is more vulnerable to oxidative stress signaling. So, and also to sort of capillary rarefaction, right? So it's such a thin sort of myocardial wall, so that pretty much sort of, you know, the perturbations that can occur in remodeling have a, you know, more severe effect. That, I mean, I'm kind of, that's sort of like me broadly, like summarizing. I can tell you, like, you know, if someone's specifically shown that, but that's kind of like how I think about it. And then when we look at the LV function in our mice, control and knockout, there was no difference. They were all normal. Granted, you know, we didn't do a TAC version of that. But I do have, there is a project that I'm working on editing the manuscript, which has taken much longer, that shows the myocyte GC1 is an important mediator of sort of guideline-directed medical therapies, cardioprotection.
Very cool.
Yeah.
That's awesome. Okay, we've got a question here in the chat. Thanks, Emily. Very nice talk. Was angiogenesis limited to the RV following PAB, or was it extended to the LV?
Yeah, really great question. So, you know, we didn't do the additional sort of histology of the LV, but given that the LV function was normal, you know, wouldn't expect it to kind of be compromised by capillary rarefaction. And I'm trying to think in human, I think in human anatomical studies, like autopsy studies, you know, that's been shown that like the capillary rarefaction in the deceased PAH patients occur just in the RV. But yeah, that's a really interesting question. Like, is there some, I guess you're kind of getting at, like, you know, is there some other sort of like, what is that, either like soluble cytokine signaling that may also be detectable on the left side, maybe in more severe RV failure, as you know, the interventricular septum goes into the LV. Good question. I don't think there's any capillary refraction in the LV, but we didn't look at it specifically.
All right. If there's not more questions, I've got one more question, but please feel free. We have a few more minutes to put some questions into the chat. So in patients that you had mentioned early on in your talk, differences in patients, male and female patients with PAH, I'm wondering if on RV biopsy, you've seen that same change in fibrosis in patients that have PAH and RV failure. Has that been reproduced in humans?
I would love to kind of do that study. So the challenges are, you know, endomyocardial biopsy carries risks, and like sort of the indications for it are really selective. And so it's really hard actually to get an IRB approved that involves endomyocardial biopsies. And in general, like, so for trying to look at sort of RV dysfunction, you know, ideally, yes, PAH patients would be the ones to really study. But they're actually quite rare. And you know, there is no indication for those patients to ever get a biopsy. So it's really hard to do any type of clinical study that involves biopsy. And so like the studies that have been done are typically like, you know, on autopsies, like using, you know, tissue collected from, you know, deceased patients. I mean, that's kind of how we were able to do our work in COVID, was just the, you know, really horrible death rates that we saw here in the beginning. Yeah.
Great, thank you. All right, well, that was a wonderful talk. Thank you so much for your time and sharing your science with us. And I'll pass it back over to Mark.
All right, thank you. So I want to thank our speakers once again for some outstanding talks, both Honor and Emily. Very engaging, wonderful talks. This is a great session to kick off our webinar series this year. And of course, I want to thank Eric and Glynis for their outstanding moderating. So I thought this was an excellent session. I definitely learned a lot, lots of interesting data. I thought the two seminars both kind of complemented each other, which was interesting in and of itself too. So um, as I mentioned, we'll be having another webinar sometime in June. So please be on the lookout for the announcement for that. And again, if anyone is interested in presenting, please feel free to reach out to me or any of the other members of our MCI committee. But thank you again to the presenters, to our moderators, and of course to all of you for attending. We greatly appreciate it, and we're looking forward to seeing everybody again soon. Everybody, have a good week.